So this is a picture of the small house I grew up in until I was 12. My elementary school was approximately a mile from my home, and I remember walking to school. There were 5 different ways to walk to school. I could not stand to go the same way during the week, even if it took twice as long. For research, I focus on different scientific questions instead of different paths to school. My research addresses how microbes in our intestinal tract impact the development of colorectal cancer through the breakdown of dietary fiber. We have a mutualistic relationship with microbes or bacteria that live on us and within us. This picture illustrates that close relationship, and we have approximately 10 times more microbes than cells in our body. In fact, this means that each and every one of you sitting in the audience is only 10% human and 90% bacteria. It's only when this relationship goes bad that you start to see diseases such as colitis and inflammatory bowel disease. In less subtle cases, changes in microbial populations are associated with colorectal cancer. Just in the past 20 or so years, research has uncovered important roles of gut microbes in several other diseases. My research deals with chemoprevention from dietary fiber. Each and every one of you has ingested fiber today. So what is fiber? Fiber is a component of our diet that's not digested by you, human enzymes. Instead, it's digested by specific bacteria in our intestine. There have been several big studies that have associated consumption of high fiber with reduced incidence of colorectal cancer. However, there have been other studies that have not found this effect. What individuals showed varying chemopreventive responses based on differences in gut bacteria, which we know metabolize fiber in the first place. Dietary fiber escapes digestion and absorption in the small intestine. Once it reaches the colon, it's converted to short-chain fatty acids. These include acetate, propionate, and butyrate. Butyrate is important because it serves as the primary energy source for the colon, and it also selectively kills cancer cells while inhibiting their growth. In fact, the chemoprevention effect of fiber has been attributed to elevating butyrate levels in the colon. In our experiments testing the effects of fiber towards colorectal chemoprevention, we sought to control many factors that could influence human results. These include using genetically identical mice, defined fiber diets, and controlling the bacteria. So how do we control bacteria? Well, we can maintain mice in sterile isolators as shown. It's similar to a plastic impermeable isolator, and it's basically impermeable to the outside environment. Colorectal tumorigenesis is then induced and prevention is analyzed. We could test our hypothesis whether dietary fiber protects in a gut microbiota-dependent way. In the first 2 isolators, we colonized mice with bacterial strains that do not produce butyrate. Then we also added an isolator where we provided butyrate directly in the diet. And then in the third isolator, we actually provided mice with those same 4 bacterial strains that don't produce butyrate, plus one that does, called butyrovibrio fibersulfins. On the y-axis, we have the number of colorectal tumors. On the x-axis, we have our different conditions. There are no differences between the 2 groups when you lack butyrate-producing bacteria. Okay? Thus, if you lack the butyrate-producing bacteria, a high-fiber diet does you no good. If you have the butyrate-producing bacteria or butyrate directly in the diet, you see chemoprevention. In addition, we looked at mutation spectrum because mutations are a hallmark of cancer development. To do that, we used a technique called laser capture microdissection, where we would cut out tissue from stained paraffin-embedded slides. And so when I would cut out a section, as shown here, I would take that section and basically I would isolate the DNA and then sequence it and do mutational analysis. And what we found was that the high fiber group not only had less tumors, but also the tumors were less developed based on the mutational analysis. In other work, I've shown that butyrate selectively kills cancer cells by accumulating inside the cells, while normally it gets broken down or oxidized. This is how it regulates gene expression in cancer cells. So the next thing we want to do is address whether butyrate was accumulating in our tumors. These peaks basically represent the butyrate levels in normal colonic tumors and colorectal tumors. Notice that it's higher in the tumor. Expanding this to our groups, we see that butyrate levels are much higher in tumors derived from mice fed the high fiber diet plus the butyrate microbe and the ones that fed the butyrate directly in the diet. So what about human colorectal cancer? What we see in mice in terms of butyrate accumulation holds true for colorectal cancer. However, there's one important caveat. That is that in colorectal cancer, patients have a large diminishment in butyrate-producing bacteria compared to non-cancer patients. And so you have to remember that a high-fiber diet had no effect on reducing tumors or stopping colorectal cancer in mice that lack this butyrate-producing microbe. So why does butyrate accumulate in a cancer cell? What we hypothesize is that it doesn't get broken down in a cancer cell where it does get broken down in a normal cell. This is shown here where the dark arrow is going in the direction of butyrate metabolism in the normal cell and away from butyrate metabolism in the cancer cell. And so how do we test this hypothesis and how would we measure butyrate oxidation? Well, we use this instrument here called a Sears XF analyzer, and it's been an invaluable resource. It's actually the only one at an academic Research One institution in all of Tennessee. This instrument measures cellular metabolism, which includes butyrate metabolism, which is what I use it for. In this next slide, basically we developed an experimental strategy that would allow us to measure butyrate metabolism. Here's a typical result showing you that we can increase butyrate oxidation as we add more butyrate to the cells, showing that it does work. And in addition, now comes the moment. So we wanted to test whether butyrate oxidation was different in a cancer cell versus a normal cell, which could lead to the accumulation. Lo and behold, what we find is the green or the purple shown here, it actually is higher in the normal cell compared to the cancer cell. And so what we've learned so far, so what I want you to take home, number one is that you need both the diet and the bacteria are important in reducing colorectal cancer. You have to have both. And so eat your fiber, be nice to your bacteria, and perhaps they'll be nice to you. In the meantime, I've actually stopped by the gas station and I got 2 fiber bars. And then I also have your probiotics, so you need both.